So today I'll be speaking about DNA replication. It begins with DNA helicase, which unzips the double helix by breaking the hydrogen bonds between complementary bases, exposing two single strands of DNA. As helicase unwinds the DNA, it creates tension ahead of the fork. Topoisomerase relieves this tension and by making temporary cuts and then repairing them, preventing the DNA from tangling. Single strands binding proteins SSBs bind to the separated strands, keeping them stable and preventing them from rejoining as they await replication. Then RNA primase adds a short RNA primer to each template strand. This primer provides a starting point for the next enzyme, which is called DNA polymerase 3. This begins by adding complementary nucleotides to the growing DNA strand. Using the existing strand as a template, it works in the 5 to 3 direction. Then the building blocks for this process are nucleotide triphosphates. These molecules provide both nucleotides and the energy needed for DNA polymerase to add them from the strand. On the leading strand, DNA is synthesized continuously because the template runs in the right direction for DNA polymerase 3 to keep adding nucleotides smoothly. On the lagging strand, however, DNA polymerase 3 works in small pieces called Okazaki fragments because the template runs in the opposite direction. Once the Okazaki fragments are made, DNA polymerase 1 replaces the RNA primers with DNA nucleotides completing the strand. DNA ligase seals the gaps between the fragments, creating a continuous strand on the lagging strand.